So now that we have discussed DNA replication, we're going to go into the next two very important steps, which are, or I shouldn't say steps, but processes, which are transcription and translation. So, flow of genetic information, central dogma, okay? Genetic information goes from DNA to RNA to protein. DNA is the genetic code or genotype, right? Which we talked about with Mendelian traits, which is why it's always indicated by two letters, right? Is expressed as proteins, which provide the physical traits, aka the phenotype of the organism, right? So we've got our genetic code, it goes to RNA, it goes to proteins, which gives me a black lab versus a chocolate or a yellow or a silver lab. Okay, so transcription is the process in which the molecule of DNA is copied into a complementary strand of RNA. Okay, transcription, one of the ways I look at it, it's the same language, it's a slightly different dialect. Okay, so we're still talking the language of nucleotides. However, we have a couple little nuances in there, which we'll get to in a moment. Okay, it's sort of like the whole pop versus Coke thing. We're still speaking English, or pop versus soda. We're still speaking English, but depending on the region that you're in, we might have slightly different terms for it. Translation is RNA into protein. Now I'm moving into an entirely different language. I'm going from nucleotide language to amino acid language. Okay, this is vastly different. This is going from English to Spanish or English to French. This is just a nice little depiction of it, right? My DNA never leaves the nucleus. I synthesize some messenger RNA, which can leave the nucleus, makes friends with my lovely little ribosome here, which then converts that into a polypeptide or a protein. So I have three types of RNA. I've got messenger RNA. This is RNA that decodes the DNA in the nucleus, and it brings the DNA message out of the nucleus into the cytoplasm. Okay, it's messenger RNA because it literally is carrying the message. Okay, tRNA is transfer RNA. This has an anticodon, which we'll go into details in a minute, okay? But it will bind with the messenger RNA because it's the opposite, right? So if I have CCG, my transfer RNA is going to be GGC, okay? So it'll bind with it. And this carries an amino acid, and that determines what amino acid joins the protein chain. Our RNA is ribosomal RNA, and this is the RNA that simply makes up the ribosome, okay? So I've got, this is going to be in my nucleus, transcription, okay? My RNA polymerase, okay? I have, I've got helicase over here. It's breaking things up. Again, this is the other reason why I want those weaker hydrogen bonds so I can get in and access this. My RNA polymerase, it goes the entire time. Instead of the DNA polymerase coming in after a little bit of that primer being built in DNA replication, now I've got RNA polymerase the entire time, and it makes my messenger RNA. Okay, so initiation, I have RNA polymerase that attaches to DNA at the promoter, and it unzips the two strands of DNA. The RNA polymerase then reads the bases of DNA and completes the messenger RNA. And then when it reaches the terminator sequence, the stop codon, it detaches from the DNA, and the RNA molecule floats off into the nucleus and eventually into the cytoplasm. I'll have you guys watch that on your own. I'm not going to do that right now. So the DNA codes tells how RNA is put together. If I have DNA that's ACCG, I'm going to have U, GGCAUUGC. This is where we're in a different dialect, okay? There is no thymine in RNA. There is uracil. I joke with the sixth graders that it likes to yell, AU, okay? I know it's stupid, but it works. Anyway, and that's our pop versus Coke, or pop versus soda. By the way, in the South, they call everything that's a carbonated caramel covered beverage is a Coke. It doesn't matter if it's Pepsi, it's a Coke. Anyway, this is our slight difference. Same language, nucleotides, different dialect, uracil instead of thymine. Each set of three bases is called a triplet or a codon, okay? Non-coding segments are called introns, and the coding segments are called exons. This is how we get all of the various possibilities that we have from only 26,000 genes, okay? When I was in college, we were told when this all was going to be sequenced, we estimated between 100 and 150,000 genes in the human genome. When we got done sequencing it, we have 26,000 genes in the human genome. That's literally less than corn. It was a bit of an ego blow, but it's because we have flexibility, okay? So I'm just gonna say this is my clothing example, right? You can take, in theory, say, I'll do ladies and gentlemen with this, right? You could take a button-up shirt, okay? You could put it with a pair of khakis and make it dressier. You can put it with a pair of jeans and make it more casual. Okay, so I have these exons here, right? I take out the introns and I get this. In theory, I could just have these two exons and exclude this one entirely. I could have this one and skip this one entirely. I can put them in various combinations, which then dramatically increases my possibilities. Translation is the process by which RNA is used to make protein then. Okay, so remember, I'm taking and I'm now going from nucleotide language to amino acid language. So in the cytoplasm, a ribosome attaches to the messenger RNA and translates its mesh into a polypeptide, and this is aided by transfer RNA. And this is what a transfer RNA is, and it sort of looks like a T. That's the other reason it's called that. So ribosomes are vice grips. They hold everything in place. Okay, it's really the purpose of them. I have a P and an A site. Okay, so basically it goes along and it reads. Okay, so for example here, I have this P, this is in the P site, this is in the A site. A polypeptide will form here. This will move over and this guy will float off and somebody new will come in. Okay, so in mRNA, a specific tRNA and ribosome subunits assemble during initiation. Okay, so I've got this methionine is my start codon. Okay, always. So, right, I've got the initiator transfer RNA. My mRNA is here, start codon, ribosomal subunit. Okay, my small one. My large one comes in here, wraps around this. I have a secondary site here. The mRNA moves a codon at a time relative to the ribosome. A tRNA pairs with each codon, adding an amino acid to the growing polypeptide chain. Okay, so initiation, the messenger RNA binds with the small ribosomal subunit. I got my start codon in the P site. The large ribosomal subunit binds to the small one. Okay, so the tRNA is now in the P site to create a functional ribosome. The anticodon of the incoming transfer RNA pairs with the codon on the mRNA. Okay, it comes in. I then have a polypeptide bond that forms between these two amino acids that are sitting there, hence the vice grip. It's got to hang out there so that there's time for this bond to form. Okay, then this guy leaves, this guy moves over here, and then I just continue down the line. Elongation continues until stop codon is reached. You do not need to memorize that UAA, UAG, and UGA are stop codons. The completed polypeptide is released and the ribosome splits into its subunits until it can be used again. This you are going to get on a test. This is very important that you be able to read. This is something that we will do in class, don't worry. So my start codon, A, U, G. Okay, that's where I go. So say I have CGU. Okay, so I have C. Okay, and then I'm going to go over to where those two intersect, CG, and then I have my third base over here, U. That's arginine. Okay, but we will practice this in class. And you are never, ever in the history, like in graduate school, I wasn't expected to memorize this, okay? Just to make you feel better, like you will never have to memorize this. So here we go. My translating genetic code here. So this is what I am trying to translate. This is my messenger RNA, okay? And then this is my polypeptide that comes from it. And the stop codon has nothing. That's why it's a stop. Okay. So mutations within a gene can be divided into two general categories. I've got base substitutions, base deletions or insertions, and they can result in the change in the amino acid and proteins. okay? So for example, sickle cell disease, I have a substitution. Instead of CTT, it comes CAT. And now instead of glutamine, I have valine. And that dramatically changes what my protein, how my protein folds. Okay, because remember, these amino acids, whether they're hydrophobic or hydrophilic, are going to determine the tertiary structure of the protein. And if I change the tertiary structure, then it can't do its job anymore. Because remember, proteins' jobs are based on their structure. A missense mutation, my altered codon still codes for an amino acid, although it may not be the right amino acid. A nonsense mutation is an altered codon, is a stop codon, and translation is terminated early. Okay, so all of a sudden I don't get the whole thing. A frameshift mutation is when I add or delete a nucleotide. Okay, and that means that I shift my reading frame. Now my codon shifts, and that can really screw things up. Although mutations are often harmful, they get a bad rap. Okay, mutations are actually really good. Blue eyes is a mutation in humans. The ability to ingest another species' milk is a mutation in humans. I don't think I'll have time. You know what? With everything going on, there's a theory out there. There's a video that I normally show called "What Darwin Never Knew." It's an excellent one. You can find it on YouTube. It's a NOVA presentation. It's really good, but it's really interesting. There is this gentleman out there who's done research, and he was interested in muscular dystrophy as a brother with muscular dystrophy. So this sort of led to the course of his research. And so he's doing research and comparing primates, including humans. All of a sudden he notices that there's this major mutation in a gene. And he's like, we should all have a big problem here. Come to find out, it is actually a mutation in the gene that is the muscle that allows your lower jaw to open and close. Okay. In a gorilla, it's the size of your quadriceps. Okay. Right. That's your big thigh muscle, for those of you who are not so hot at anatomy and physiology. He actually thinks that this mutation is part of what has led to skull change, because this huge muscle makes gorillas' heads, their skull, more constricted. In humans, it's not as constricted, which may have helped lead to the formation of our prefrontal cortex, which may be part of the evolutionary steps that allowed our brain to develop the way that it did. It's just really cool if you think about it. Okay, we used to have short-necked giraffes. Okay, they don't exist anymore because of a funky mutation. So this is just going to put it all together. Okay, I've got my messenger RNA. I'm reading 5' to 3' here. Okay, my ribosome is here. My transfer RNA comes in. Gotta love the sounds. Okay, I've got my other one. I have this polypeptide bond form, or this peptide bond form rather. It moves on down the line. Now I've got a new one. It just keeps moving on down the line. Okay, and that